So very good afternoon to everyone present here in this afternoon. It's my great pleasure to be with you all, to be along with the Babatlan College of Pharmacy to present an very interesting topic about the pharmacophore modeling, database creating, a kind of a lead optimization technique in a ligand-based drug design. So there are a lot of in silico works or a computational chemistry work or chemoinformatics work can be carried out using in various tools and softwares. However, today I'm going to present to you. Is my voice is audible to everyone?
Ma'am,
Ma'am,
Yes,
your
thank
video
you.
is not appearing.
No, sir. When I put a video, the things it'll take a the slides will slow very more
No
slowly.
problem, no
That's
problem. Go ahead.
Thank you so much. So pharmacophore modeling is a most, and I could say that a crucial technique for the people who is working on a different background, like pharmaceutical chemistry, even biotechnology, or chemoinformatics, medicinal chemist, organic chemist. So wherever you are seeing an atom or a compound, this pharmacophore modeling can be implemented. And if you are interested in a drug discovery, or if you want to develop a new lead compound for any kinds of a disease, I would say that this is an appropriate tool to perform your operation to screen or to take in a promising drug targets or a drug candidate for your experimental analysis. As I am going to take you along with me as I work on slides, you will gain more knowledge about what is a pharmacophore, why we need to do pharmacophore, what is its important and biological significance. Let's work on slides. Before that, let me explain about your company for a while. Alton Technologies is in a most from a decade company, is involved in a fingerprint and various technologies. And one of the imprints in the life sciences is a Bioware Discovery Studio as a part of Dassault Systems. And we are also now stepping into another technique called 3D bioprinting. We are the first people who is getting into that bio 3D printing. Instead of a 2D printing, now 3D printing of any biological material can be printed. That is going to be our next evolution in our market. And now we are into 3D Bioware Discovery Studio, the software as a compute solution for your computational biology and chemistry. So you can see here our awards, and then we are alliances within various software companies. Moving on to the Bioware Discovery Studio. Bioware Discovery Studio is previously called as an Accelerate Discovery Studio software. And now it has been taken over by Dassault Systems, so we call it as a Bioware Discovery Studio. This Bioware Discovery Studio have a rich scientific portfolio, starting from a unified lab management, process productions, quality and regulatory management, and also into the collaborative science. So what is it unified lab management? Every time when we get into a laboratory, we will plan certain things, and then we'll go for an certain experimental analysis, and finally we'll be writing in a lab report. So for that, we'll be taking always depends upon the paper and pen that has been replaced into a technique called an electronic laboratory notebook. So wherever you are doing experiment today, and after 10 years, you want to access your experiment and what standard operations you use, what SOP you introduce. If you want to analyze and you want to integrate and you want to develop a new protocol from that, you can access via your cloud computing to a system or to a software called electronic laboratory notebook. And process and production operations are a primary part of this, and more into the quality and regulatory management, where you can see the quality of we start from a documentation, processing, and compliance and collaboration. So the collaboration science, right from a predictive models, even if you have a linear sequence which you get a model to get a predictive structure of the particular protein or enzyme or any kind of a drug target, it is now very easily can be done by using a software called a modular software, which is a part of a Bioware Discovery Studio. And no, I'm interested into the macromolecules or the biologics or antibody predictions. You can use an antibody sophisticated tool that is also a part of the software where you can identify the novel therapeutics. And some people will be working on drug delivery or engineering the materials. For those people, we are having a material studio software where you can engineer the materials and you can use it for a drug delivery purpose, like a polymer-based drug delivery, carbon nanotube drug delivery, or any kind of metal-based drug delivery. So all those things we can able to perform using a software called Material Studio. And if people are interested into the agriculture chemicals and formulation or a cosmetic chemicals and formulation, starting from an herbal products or herbal things, that formulations also you can able to do, and agrochemical screening also it is feasibly can be done with the toxicity free chemicals using a discovery studio. So we have a sophisticated tools which is built by using a validated purpose and approved by a food drug approval with a food drug administration, and from the national toxicity program we have certain compounds and then approved things, and through that we can predict whether your molecule is going to have an toxicity, irritancy, ocular irritancy. So all these kinds of analysis can be carried out using an TopCat or ADMET analysis tools that is a part of the discovery studio. Let's move on. Collaborative science. This collaborative science, we have a comprehensive scientific data analysis and then sophisticated tools to perform this analysis actually, right from a life science predictive analysis, as I told you, even material science, scientific pipeline design and execution. No, I don't want to introduce or want to use the softwares that the people want to do. Rather than I am interested in designing a own pipeline for my research. Of course, if you're interested in that, we have one more tool called a pipeline pilot, where you can program using a scripting languages and introduce here and build the component and pipeline your process till the finishing of your identifying a lead compounds, you can able to pipeline your research. And of course, we are also into collaboration, externalization, cloud computing, and various research applications and chemical inventory management, and more into your bioactivity and chemical sourcing content. Moving on to the little bit depth into it, what we'll be doing in life science predictive analysis. The life science predictive analysis, we are more into biologic modeling and simulation. So if you model some kind of proteins, you have to see whether that model will be behaving as like an in vivo system or not. That you can study by using a technique called molecular dynamics and simulation. Same thing, you can apply the technique to the small molecules, where the small molecules are very important for your drug discovery and developing the lead for your experimental analysis. And QSER, it's a most important and the familiar technique with the people who is working on a pharmaceutical background. It's a quantitatively predicting the structure activity relationship. We have more than, I could say that we have regression models, classification models, and then 3D models to develop your QSER. And these all tools are put together in a one platform called a Bioware Discovery Studio. So today I'm going to explore you with how do you get or how do you perform this Pharmacophore modeling using the software, and what kind of routes or the trials you can get into it to come out with a good lead compounds for your experimental analysis. That is going to be objective of this particular session. Okay. Anyway, if you get into it, we should know why we need to do drug discovery or a computer-aided drug designing. So if you have these kinds of inquiries in your mind, then definitely you should come across two things, either the protein or the ligand. See, what is this ligand, compound, and molecules? All denotes the same notations, but the way of people representing or pronouncing or being acronyming will be varying. So if you have your known protein and your known ligand, that means everything is steady. I have synthesized my molecules. I know for which drug target I'm going to use. Maybe a cancer's or Alzheimer's or Parkinson's, whatever is it. Then that technique we call as a structure-based drug designing. Say for instance, on the other hand, you have your known proteins, like in COVID-19 situations, there are lots of repository for your PDB structures. If that is the case, you have a lot of protein structures or in a protein data bank, and we don't know which small molecule is going to effectively work on this particular disease. Then if that is the case, you have to move on to a technique called an de novo drug designing. So that will help you to identify the which fragment or a parent molecule is suited for this interaction, and from there you can introduce a fragment-based drug designing And using that technique, you can build your links and fragments to come out with an excellent drug for your further users. And let me see and put into word, if you have a known ligand, but you don't know for which disease I have to use it. And if that is the case, you have to go for a ligand-based drug designing or a pharmacophore. So we are going to fall today into this technique called we are having a known ligand, but we don't know for which system or a receptor or a protein or enzyme that we are going to utilize it. So let me get into an extensive or an odd say case where we don't know anything. See, I don't know anything about it, but is still interested in doing this kinds of computer aided drug designing. So what will be my solutions for that? So final first, I will be starting with the literature study and rational screening with a library design analysis. Then I move on to the any one of the technique for which is falling under category of a SCAD. And impact of your drug discovery is a bioinformatics. So it starts from a gene product is protein put together. You can study in a platform called bioinformatics. Little bit moving inside it, you are studying the structure of your protein and how it is behaving and binding site. That is called a structural bioinformatics. And still moving into a ligand part, lead, hits, compounds. Then we call as a chemoinformatics. Put together protein along with the lead, we call as a structure-based drug designing. Or else if you're leading, you are linking in the lead where your lead molecules are obeying all the pharmacokinetic properties, then we call as the area liquid modeling. So all these kinds of techniques or methods will help you to screen or be key steps for your drug discovery process, combining or interdisciplinary working with the various domains will accelerate your research process and help you to come out with the best solution to your drug discovery process. That is actually wherever, whatever we do, finally we come out within a drug candidate. That is our major aim for your any kind of a drug discovery. Why LBDD? What is a ligand-based drug designing? What is it going to do that? So then we will do that. So it should be always raised in your mind. I should not blindly take a structure-based drug designing or a ligand-based drug designing. When certain problem statement is arising, where I have to put this results, where I have to start, how do you start? So if that is the case, you have to think about it. Then I can apply this ligand-based drug design. So then it will work out. In the absence of your label protein structure or any protein structure information, then you can introduce your ligand-based drug designing. It is a trusted area and awareness of identifying new ligand molecules that will bind to your specific drug targets. Indirectly, the drug designing in a traditional organic synthesis by a medicinal chemist, even 3 to 4 decades before also, they introduce a ligand-based drug designing without knowing that that is a drug designing. They use some parent molecule. From the parent molecule, they have been derived lots of derivatives, and they tested all the derivatives and with the parent molecule to see the activity relationship and whether that particular compound is curing or not. So from there, very few decades before itself, we have started doing a drug discovery process indirectly. So chemical compounds or the starting point, or he is a hero here for ligand-based drug designing. So let's see about how this started. So if you take about it initially in traditional ligand-based drug designing, the target and the diversity-oriented organic synthesis has been playing a major role in that discovery process. In 1960, the systematic method has been introduced and named as retrosynthesis. And this problem has been taken towards identifying key elements in a reaction and rather than reacting substrates and forming synthetic routes and transformations for that. And repetitively, they introduce a synthetic chemist to start working on structurally complex targets and identifying a simple compound to be used for synthesis. Because synthesis is not an easy task. We have to identify the proper route and reactive points, and then once the reaction is carried out, how much yield you are getting from the yield, whether the residue is being more or not. So all these things has to be need to be analyzed, and that has been previously done even in your 1960s also, without knowing that that is actual drug discovery process. And this mechanism based or a diversity based diversity oriented synthesis has been evolved, and finally the mechanism target identification is reached for a drug discovery in a cancer research. So now going to what are the new intervention approaches you can take it up in your ligand-based drug design. So these all are the different approaches that you can do it. If you have your known actives, what is this known actives? These are nothing but the ligands. If you have your known ligands or an actives or known. Active in the sense biological activity. If you know your biological activity or don't know the biological activity, don't worry about it, because in ligand-based drug designing, chemicals are hero. When you know have a chemicals or hero, then he can do the wonders with the proteins also. When you open your protein, that is also a chemicals. That is not, it is again, it's coming as a chemical amino acid. If you think about the amino acid that is made up of a CHNOs, there also chemistry is playing a major role. So atoms, molecules that put together only forms any structure biologically, or if you want to see it in a chemically, that's called as a compound. So they are the heroes. Atoms and chemicals are heroes in our world. That is doing wonders in our, even in our biological systems, even outside the systems, and doing around the systems also. Say for instance, if you have a known biological active molecule, what you can do that? Say, let me say that there are some drugs, I could say A drug. A drug is very effective, is working for some kind of cancer, and the B drug is slightly working, and C drug is almost failure. So if I want to synthesize or come out with a still more active molecules, maybe 1 nanomolar is good for that particular molecule, for A molecule. I want to still have a good biological activity. What I can do that? So instead of before synthesizing, I can do the similarity or substructure search that will help me to identify what are the similar like molecules are present. If there is sharing, that what are the structures are sharing. So that is one of the way where you can study the similarity or the substructure search. Or else, I know only the compounds, I know the activity values for the compounds. Maybe I have hundreds of compounds in my hand. I don't want to do my activity for my new synthesized compounds. Then you have to get into a technique called quantitative structure active relationship, where we are trying to build a model. Using that specific model, you can screen your known or unknown activity, known or unknown molecules, or even can screen a database of molecules. And moving on to the pharmacophore modeling, pharmacophore modeling is also a very important and then most fruitful technique, I could say that, because all these things in QSAR or similarity search, we are dealing only with the 2D spatial arrangements. Only in the pharmacophore modeling, we are going with the 3D spatial arrangements. Why 3D is more important? Because we are also 3D systems. So if we do the certain things in 3D spatial arrangements, then we also believe that that will work out very specifically in a 3D model, human models also. That's why the pharmacophore modeling is a most promising technique for screening your ligands and compared to a 2D based tools. And say for instance, I don't know anything about it, but I still want to know that, then you can go for a machine learning algorithms and you can predict the activity of the molecules, and then you can come back to any kind of approaches. These all are the LBDD approaches is playing a major role in your ligand-based drug designing. Approach number one. So now I'm trying to solve and show you how to approach this for case number one, case two, and then case three. And if you have this one, how to analyze, how to get into it. That's what we are going to discuss now. Let me take the approach one. So how do you do the substructure or similarity search? The approach one, it's a molecular similarity. That means you have two molecules in your hand. You are drawing or sketching at two molecules. How do you say they are both are similar? We have to do some kinds of an analysis or a matching technique to come out to know how similar the molecules are. It will be widely used concept in your rational-based drug designing. And moreover, this is a basic principle to analyze whether structurally similar molecules will have a same biological activity. That is one hypothesis today. If you take one structurally similar molecule, maybe a compound which is containing a phenol ring connected with an naphthalene ring, and then NH, OH in a place, different positions, and we are trying to see the similar like molecules, maybe, but position is very important, but slightly similar molecule will behave like this molecule. That is an importance of introducing a molecular similarity. That means coming out and searching as functional similar molecules, which is common aim in your drug designing process and could be accomplished to search for similar molecules. See, what could be a similarity? It could be a similarity based on the properties, maybe a log P similarity, molecular weight, hydrogen acceptor, donor, or you can say that maybe a log D. Whatever the properties are there, even you can search via two properties or by looking into the fingerprints. What are these fingerprints? Fingerprints are nothing but a chemical signature of your molecules. That will say these are signatures, CNHO, connected with double bond O, and then balanced with the NH. So that is one fingerprint for a particular molecule. So wherever the fingerprints are going to present, pick those molecules. That's what the people will do in your various databases like a Zinc, as well as in a PubChem, Asnix, ChemDiv, Chembl. So lots of publicly available databases are also available to do this search. But here in a Discovery Studio also we are having these kinds of a search to identify your structurally similar molecules. And mostly it will be between it. I try to identify the distance between the structure and all your physiochemical descriptors in a space. And apart, there is also another thing. One is by means of other by means of a molecular is first, and then is by means of numerical properties. That means if you're giving you're measuring in terms of a distance, we'll be studied the distance between the two positions in your school days. The same thing we are applying here indirectly, but we don't know where to apply this. If you have two structures, how do you say this is a reference ligand? This is an other ligand. You can calculate it by means of a position of your molecule or numerical properties that can be measured by means of an Eleutherian, Manhattan, 1 minus correlation, 1 minus correlation square. These are different distance metrics. It will be helpful for you to come out with a numerically similar property of your ligands. So how do you do that? If you're having a certain distance formulas will be applying. Once it apply a I and J positions and try to calculate the distance and you get that. So how do we implement this in production? I mean, in terms of working. So first you have to get into, say for I am having a bulk database of compounds from external resources. Maybe I downloaded from Asnix or ChemDiv. My compounds are get ready with me. I have reference ligand. I use in hydroquinone drug so as a reference ligand. What I will do in the sense, in the small molecule table, I have a protocol called find similar molecules by numerical properties. So once I select that particular protocol, I have to give as input ligands. It's nothing but your database of molecules. And reference ligand is nothing but your molecule which you want to be a reference, like that you want to select the data compounds or database. And what to find, it will ask you percentage of more similar molecules, what should be the percentage. You can decide the percentage. And using a distance, which distance you want to perform, Eleutherian or Manhattan, whatever distance, you can measure that distance. And then if you want to select the means of user properties, you can give that option also. Once you select, just simple click, but when you run this, we call this a protocol. Once you give the protocol, you get your results like this. It will show you the description, how long it has run, and how many molecules are retained from your database. So when I try to give 500 molecules, out of it 129 molecules are similar, and the name will be displayed over here, and what is the distance, and compare to that the reference. With the reference, how much is the distance? How much is similar is it? Is it similar? Yes. How much similar? Yes, true or false. All those details you can able to get it from here. True or false. It is very easy to interpret also. True means yes, it's a similar. False means it's not similar. So I can take only the similar molecules Based on the distance, then I'll be taking for a further analysis. This is a one way of screening of molecules based on numerical properties. And similarly, by means of fingerprints, in the fingerprints, we are having various kinds of fingerprints based, that is by means of Tanimoto, dyes, cosine, target. So henceforth, hereafter, when you want to search or similarity search in any kinds of database, look for that, whether that particular tool is using which kinds of method of similarity, whether it is using a fingerprint based or numerical based. So when you learn about this one, this will give you the clear picture of your idea. Whether I find when I write a paper, yeah, I selected the molecules based on Tanimoto similarity rather than telling that I selected the compounds based on similarity such as present in a specific search engine. You can be very specific when you know about these kinds of similarity measures. So how this fingerprints will be measured, I told you, right? So each and every one will be considered as fingerprints, OH, as considered as in a bits value, 0 and 1. And whether this fingerprint is matching, then that will be named as 1, 0. Like this, it will try to identify the bit by bit and with the fingerprints of your molecule, and then matches and shows your results. So this is how the SA and SP is nothing but a positions, number and the bits. It is present in the bits are nothing but 0 and 1s. Once a hit, okay, when I find this fingerprint 1, I never find the fingerprint 0, 1, 1. Like this, it put together, it forms a values. That value will be converted into one scores. That scores that we are interpreting for a further analysis. So how do we do that? Same inputs. I'll be taking a database of molecules along with my reference molecule. But here I'm going to call it the all molecules with the minimal similarity. What should be minimal similarity? What coefficient I want to take, Tanimoto or a dias or a cosine, what I want to choose, and which type of fingerprints you want to select. You can select that fingerprints, screen in the database, you get the results like this. Whether they're as like a reference molecule, what are the molecules are more similar. And each and every fingerprint based also you can able to get it. So this is the first approach of identifying a similar like molecules in your ligand-based drug designing. Now moving on, we will be getting into one QSAR. This is second approach. It's nothing but a quantitative structure activity relationship. We also call, there is another technique called quantitative structure property relationship. When you get into the QSAR, you have to address yourself into two different questions. One is, what feature of molecule is affect its activity and what can be modified to enhance the properties? Say for instance, you are having certain molecules on your hand. You have one molecule and you know the activity of molecule is good. But if I want to enhance a biological activity, what I should do, where I have to alter, which position I have to give the reaction, whether reaction will go in this position or not. All these things should be keep in your mind and keep on, it should crack in your mind to ask about the QSAR. We cannot do simply the QSAR because it is a statistical and mathematical model to be analyzed with your biological activity to show whether that molecule is active or not. So you should have an inside biological process to change the position of your molecule, and based on changing the position, whether that is enhancing or it is a decreasing its activity, it's all based on the position where we are introducing modification. And then it's just basically what you have to know. We should have a dependent variable. It's nothing but your always your biological activity value. Or if you want to go into independent variables, those are the descriptors or the independent variable. The independent variable could be a fingerprint or topological descriptors or electronic descriptors or A log P. What are the properties of the molecules we consider as independent variables. So we try to have a correlation and regression between them and identifying which QSAR of the descriptors is making a good sense to correlate with your biological activity. That is an idea of introducing a QSAR. So consideration, dependent variable, I told you, right? So experimental value is considered as a dependent variable, and independent variable should be a choice of a descriptors. So when you try to choose an independent variable, you have to be very careful because your independent variable could lead you for a poor QSAR model. And some kinds of parameters that you try to correlate will have an overfitting. When you have this high accuracy or training set of compounds, will have a low accuracy test set compounds. And you will have think in your mind how much molecules you must be in training set. Minimum 20 to 30 molecules in a training set. And then if you have a diverse molecule, that will be appreciable because that will be a very good QSR model. Now, moving on to the third approach. It's our actual approach. What is a pharmacophore? So, what is this pharmacophore is going to do essentially in a biological activity? What is its role actually? In 1909 itself, Ehrlich has defined the framework of a molecule that works essential features or a crucial functional group of a molecule which is responsible for a biological activity is called as a pharmacophore. So when you remove that specific functional group and specific position will lead to an inactive molecule. So that is called as a pharmacophore, the functional group of a molecule responsible for that. And a pharmacophore, we call as if you ask a cheminformatics, they will tell like this. It's the ensemble of steric or electronic features that is necessary for your interactions with a specific biological target to trigger or block. Either it should fight and kill that molecule, or it should go along with that molecule and enhance the process. It should be agonist or should be agonist or antagonist. It should fall in either of this category. Then only we call as a pharmacophore of the molecule. So when you have your ligand, so in a discovery studio notations, we have different notations for different pharmacophores. Hydrophobic, or called as this form, you can see sign in color, and acceptors will be in green in color, and donors will be in pink in color. And if you have a molecule with a positive ionizable, that will be shown in red in color, and negative ionizable in blue in colors. So it should have a complementary pharmacophores. Acceptor is present, donor should be there. Donor is present, acceptor should be there. In case donor and donor, that will repel. Acceptor, it will repel. So your ligand should be in such a way, it should correlate or should be complement to your receptor pharmacophores also. So now we can come to know about receptor also has a pharmacophore. What is that pharmacophore? We called as a structure-based pharmacophore of your receptor. So ligand alone doesn't have pharmacophore. Even your receptor is also have a pharmacophore. And what is the use or significance of using the pharmacophore? It will help you to explain structure-activity relationship of your molecule in a three-dimensional interpretable model. And it could be a qualitative or a quantitative also. Even if you don't know about your activity value, but you still want to know your structure pharmacophore, yes, you can do it by means of auto-pharmacophore generation or in a feature mapping or by using a common feature pharmacophore generation. These are the tools that present in the Discovery Studio. And it also helps you to predict the activity of new ligands. Say, for instance, I'm trying to do the 3D QSR and I have a database of molecules in my hand, which are other, maybe after a decade, other person wants to do that. But already I finished with my predicted activity value and my ligands predict, so I have good pharmacophore. So if that is the case, I don't do the again pharmacophore based analysis. Rather, I take the already generated pharmacophore in my laboratory and screen the database of molecules. From that, I will draw the conclusion that these molecules can be active for the specific disease or not. So it will help you to design a new building blocks for your novel drug lead candidates. Scaffold copy, introducing some scaffolds into that, or there's a query database for your similar pharmacophore patterns activity. And I think in future, like a ligand database, there will be a pharmacophore database also will be there. So when you want to screen specific drug targets, you don't need to worry about it in future. Some people will generate a pharmacophore and deposit it. So you like PDP structures, you can download the pharmacophore and you can screen with your ligands and you can produce your results. So in future, these things also is feasibly is going to available. And then when you get into typical pharmacophore, we have different perceptions. So if you have things in terms of ligand perception, it's a training set selection and configuration and confirmation. Confirmation is very, very important when you come across a pharmacophore. And then how do you do that? They can be done by means of diversification. These are the protocols present in Discovery Studio, and what are the prescriptions or been displayed over on the left hand side. Ligand perception, you can go for a feature mapping or a diverse confirmation of ligands. In terms of pharmacophore generations, you can go with an qualitative based, quantitative based, structured based, or a refinement with an inactive ligands. So what are the tools we can use that for that? So for a qualitative analysis, we go for a common feature pharmacophore, and quantitative, we go for 3D QS of pharmacophore, and structured based, we have an interaction generation. But in the 2020 version of us, we are having even the interaction pharmacophore generation that will help you to develop the structure-based pharmacophore easily. And refinement of the active ligands, we are having a steric refinement of the excluded volumes. And analysis. Analysis can be done by means of ligand pharmacophore mapping, pharmacophore comparison, screen library, ligand profiler. This ligand profiler is an excellent tool for an computational chemistry, will help you to identify the multiple drug targets with the multiple ligands. So far, we are doing only the one drug target and then multiple ligands. But think about it, multiple drug target screening at the time with the multiple databases can be handled through one specific protocol called a ligand profiler. And prediction. So from the prediction, we'll be estimating your activity and identifying your potential leads. So that can be done by using only your pharmacophore, these tools. So what is basically required for this? You should have ligands, you should have a pharmacophore that could be a qualitative or quantitative or structure based. So any one pharmacophore should be there for you to perform to identify the activity or predicted activity for your molecule. So now let us briefing into the pharmacophore modeling. In the pharmacophore modeling, if you know the biological activity value, that's why I told you. If you know biological activity value, you can go for 3D QSR pharmacophore. If you don't know the biological activity value, you can go for auto pharmacophore generation or your common feature pharmacophore generation. So let me describe, because anything, if you want to predict by means of quantitatively, that is more important for an analysis. So that's why I just picked only a 3D QSAR. That is very important. Why? In the sense, when I try to build a 3D QSAR for one particular disease, maybe very target specific, which 3D QSAR is very target specific. I could say that I am trying to develop the drugs for an Alzheimer's, maybe an or and I could say that coming to an breast cancer, estrogen receptor. Okay, there is an estrogen receptor. In the estrogen receptor, we have alpha, beta, and gamma. Maybe I could pick an alpha. In the alpha estrogen receptor, I try to design a pharmacophore for the specific. So what I will try to do that, I will try to check from various databases like a binding DB or Campbell or any other databases, which is going to have an activity value, or from the literature work and try to select the compounds and the respective activity value has been taken care. So put together, I am forming one particular compound database, and I'm going to use that as an input for my 3D QSR. So it is a very specific, it's an estrogen receptor alpha target, and the ligand is also been predicted only for that. So maybe hundreds of ligands I have been collected, I'm been ready for the pharmacophore model. I have to do this process? What is the necessary of this? Once I do this process and develop the 3D QSF pharmacophore, and I have a database of molecules, maybe my junior is going to do that research in future. He don't want to do that process. He don't need to develop 3D QSF pharmacophore. He can take my pharmacophore and screen with the synthesizingly or being synthesizingly sketched a molecule. From that one, you can pick only the, instead of going through the experimental analysis with the 100 molecules or maybe a 40 molecules, he can narrow down to 5 potential molecules with the predicted activity value when a nanomolar or millimolar. This will help him to predict the activity value. From there, he can go ahead and he can finish his research compared to a first person. He finishes research as fast as possible. That is the importance of introducing a 3D QSR pharmacophore in the ligand-based drug designing process. So how it works actually? So it uses an algorithm called catalyst hypogen, which derives a SAR hypothesis of the set of known active ligands to the given biological target. And it will be typically different from a QSAR. In the QSAR, it will be dealing only with the properties, but in the pharmacophore, it will be dealing with the molecular features in the 3D spatial arrangement. That is the difference between 3D QSAR and then QSAR. So how do you generate it? So it generates by means of a set of ligands, and hypochain will help you to identify the set of active molecules and what are the inputs we need to give for that. You should give an activity value that should be a 4 orders of magnitude. If you're giving a logarithmic value, you have to keep 3. And any kinds of an experimental analysis, there will be uncertainty will be there because of physiochemical parameters and manual errors. So that's why we also introduce the uncertainty when we want to do this program. So we introduce the uncertainty from a level of 1 to 3. You can give any levels to identify to predict or build your 3D QSF pharmacophore model. So you should know about how this algorithm works. So it's nothing, it's also a mathematical algorithm, but in the chemistry. So first of all, you try to construct it. If the constructive phase is similar like in a hip hop, it's another algorithm. So it will identify and pharmacophores that are active molecules in the training set. Once it identifies the active molecules in the training set, now the program will move towards the subtractive phase. Say for instance, there are 4 pharmacophores are present in my systems. The 4 pharmacophores, let me annotate to you all. I think that will be very easy to understand that. I don't think so. Annotation. Sorry, I cannot annotate that option. It's not available. Okay, fine. Let me say that you're having one particular molecule. In that particular molecule, 3 features, let me say very easily, 3 features. Acceptor is present, donor is present, and hydrophobic is present. And inactive molecule is trying to match with my active molecule hydrophobic region. So in the subtractive phase, what it will try to do, it will try to delete that particular feature. So it removes the pharmacophore from a data structure that is not likely to be useful. Or it inspect with the pharmacophore in a constructive phase. In common, in any inactive molecule is going to match with that, it will remove that molecule. That is what it will do in a subtractive phase. And once the 2 things is finished, then it moves to the optimization phase where it will try to do an assimilation annealing. By doing a simulation annealing, you will be getting enough to improve the scores. The algorithm will try to improve the scores, and that's how you will be interpreting your results. So it will try to obtain small permutations of the pharmacophores in a constructive and subtractive phase and help you to improve your score of your hypothesis. So any hip hop algorithm or hypothesis algorithm, this is a step in any pharmacophore. This is a method must be followed. It should construct, it should subtract, it should optimize. Moving on. So how do you do that? Say I'll go to the Pharmacophore tool, and then in the Pharmacophore tool or protocol, I will try to select the create Pharmacophore. In the create Pharmacophore, I'll opt for a 3D QSR Pharmacophore. So pretend that I collected the compounds with biological activity, and the process with more than or 30 will be a fine. And I will try to divide the test and training set in a random fashion and repeat the process till I'll get a maximum regression value. And this is how the setup will be there. So I'll try to input my ligands and activity value and certainty. And if it's logarithm, I'll keep the logarithm true. And I go for a different kind of confirmation method and look for a maximum energy threshold and features. What are the features or should be present in my active molecules, and how many pharmacophore and interface distance between your pharmacophore. And this is a technique, it's a multi-optimization technique. Okay, that means you have to keep an eye on all the parameters. One parameter may lead to your regression score less, or where it is going to work. That is in a research that we'll be doing in a 3D QSR Pharmacophore. And in case if you wanted the cross validation here itself, you can input your test set molecules and then you can introduce a leave one or cross method here, lock method here by introducing a omitting feature -1, and you can predict your results. No, I still want my pharmacophore to be statistically validated. Then even you can introduce a Fisher validation, one of the statistical parameter method to validate your pharmacophore using this kind of options. So 98% or 95%, 90%, you can give it and then you can predict your result. So this is the XML setup for your 3D quiz of 3D quiz of pharmacophore protocol. So once you do finish, okay, why do finish my process? Now we get a very detailed results. So I get a molecules, I will get a detailed result that which says me hypothesis 1, 2, 3, 4. Okay, to try to predict more hypotheses. That is good. And how do I interpret my results? That is a big task in the prerequisite of pharmacophore because it's a multiple optimization technique. I told you, right? We need to analyze a multiple parameters into it. So if one parameter is not giving you the best result, then what you have to do, you have to repeat this experiment. You have to start from the beginning. You have to see where the problem in a training set molecule or test set molecule, but make sure that your training set should not have an accuracy. If you have overfitting in your training set, that will definitely will affect your test set molecules. Okay, fine, I got a very good regression. R is equal to 0.99. Okay, then if you get like this, then definitely it cannot be compensated by your test set. If you're getting your test set 0.8 and training set 0.79 or near to that, that would be good set for your PDQSF pharmacophore model. Rather than the people are in a different ends, they both should be in near places. That is more important in your PDQSF pharmacophore. Okay, let's move on and see how to interpret the results. You get a result like this that shows we have a two theoretical costs. Fixed cost, null cost. As the name says null, the pharmacophore is the null over here. And fixed cost, that means it is fixing closer to your value, more statistically equivalent to your pharmacophore. So any of your values that should be near to your fixed cost. You can see that these values or near to your fixed cost are away from your null cost. This is the first step of your 3D QSR pharmacophore interpretation. And your written hypothesis, you saw the hypothesis in previous page, right? These are the hypotheses. Here you will get an RMS value, root mean square values, and correlation. Correlation must be high, RMS should be less, and then you should get in a configuration value also should be equally contributed to that. And you can see here for this total cost is 56. So that's what we are trying to explain over here. Cost values for optimized should be lie somewhere between your two costs, closer to your fixed cost and away from the null cost, the more significant your pharmacophore is. And if the difference is greater than 60, the difference between this cost is greater than 60, when you try to predict, that is considered an excellent model for your true correlation. What if the greater than is if it's between less than that? Still, we say that if there is a hypothesis between 40 to 60, it's a higher probability of a 75% to 90% to be accepted as a true correlation. As the difference became less than 40, then the true correlation is reducing rapidly below 50%. So this is a very important parameter that we should analyze when you want to interpret your 3D QSF pharmacophore. Moving on, we get the results like this, seeing that the maximum fit and total cost, RMS, and then you'll be seeing the molecules with an status, or what is the estimated activity value, what is the error. So all the things you'll be getting as a printed results, okay? But we need to interpret the results. That is the most important task. Let me say that I'm getting hypothesis 1, 2, I'm getting like this, 1 second. I'll get a hypothesis, up to 10 hypotheses I'm drawing. But however, if you look into it, there are some features or mapping, it's very important that we should be key on it. I'm taking these two as active molecule. And I'm seeing one more thing is called star over here. That star is nothing but it is also mapping with the inactive molecules. See the inactive molecule star. So that means every time it is not necessary, you should take the hypothesis 1 for your screening. Rather, you can take even hypo 2, 3, 4, 5 also. Make sure that it is not matching with your active molecule mapping. So I told you not only a null cost, fixed cost, even your mapping is also very important here. So your hypothesis mapping and its correlation is matters a lot when you come to your hypothesis predictions. And based on that, it is one of the papers published, which is trying to show the pharmacophore mapping and then statistical analysis for the 10 top scored hypothesis. And you get a hypothesis 1 where you get this value. You can see it in here itself, total cost, null cost. From there, they will try to put a table like this, and they see the difference between the cost. Okay, the cost is good, and RMSD is be expressed in terms of angstroms. And the correlation is good, and features. What does these features mean? Accept charge and hydrophobic aromatic, ring aromatic, and the donor. And the resistance will be calculated from a null cost to fixed cost. If the null cost is fixed cost. So, and they found the 10 statistical scoring matrix for your training kit. Similarly, you can generate a for test set also. And you can see the what is the results between your hypothesis 1 of a test and a training, and you can correlate it. Either way, either you can repeat the experiment or you can go for a mapping steps also. It depends upon the perspective of the people research that you have to choose in different methods or a combination of more than one method in the pharmacophore modeling. So what do you mean to call as an active molecules? The active molecules are nothing but which fits perfectly matching all the pharmacophores. They call as a maximum fit or active molecules. And then you can, when you want to select your molecule, your output will look like this. So you can select one by one. So this is active molecule. Why I say I cannot blindly draw the conclusion this is active. Only when the molecule is mapping within all the pharmacophores, positive ionizable, ring aromatic, and acceptor and donor. So from here you can see this. These all called vector features. What is this vector feature? That means the distance from this place to this place. If your receptor is going to lie over here, up to this distance at a various angle, it could have an interaction with your molecule. Make sure that if it is acceptor, the other molecule of your receptor atom must be a donor. If that is the case, up to this distance, it could have an interaction. That is the importance of having a vector-based pharmacophore. And these are the point of spear-based pharmacophore. You can see that. And it is also denoting the specific atom, which atom is because of that which is making a pharmacophore. And replacement of this particular atom in this position, whether it will altering a pharmacophore or that's all in your hand. You can change, you can play, you can identify, and you can draw a conclusion. It's all the computational works. You can play very well, and you can come out with a very interesting results. And moderately fit molecules. That means one of the pharmacophore is missed. You can see here, this pharmacophore is missed, and it is not matching with that, and it has been finished. So if that is the case, it has been not matching with the one of the pharmacophore we called as a moderately fit molecule. Inactive. Inactive in the sense it might be missed. See here, these two pharmacophores are totally missed out. So that's what called as an inactive pharmacophore. So like this, you can map and you can study and you can draw the conclusions. That is, which is a pharmacophore having which molecule has been high active molecules, which molecule is a moderately, and which molecule is in a moderately active molecule. So this is a pharmacophore model, and mapping will help you to understand from that. Okay, from here I'm finishing? No, it's not like that. This is just a beginning. You identify the pharmacophore. What should be my next step? Where I'm going to use this output? How are you going to predict it? That is a big task. And how do you analyze? That's as work on slides, you can know about it. And here it says even that your molecule and its binding, and you can see the confirmation, and you can see the what is a fit value, and then you can also see about which atom is mapped and which feature is mapped in 20 in your newer versions. We can even see then which atom is mapped within which particular pharmacophore, and replacement and specific mapping is also will be there. So if we say that it is an active molecule you have been given and which confirmation is taken and the ninth confirmation, I will try to predict the estimated activity value of a fit with your pharmacophore. What is a fit? So here we interpret in terms of fits. So fit maximum the fit, good the results are. If you have a least fit, the results are really not good. So that is how we have to interpret the results. And the fit is the most important thing that we have to analyze during a pharmacophore based drug designing. Moving on. So if you get output pharmacophore, we have to map the pharmacophore like this. And a hydrophobic you have to match like this, and you can do this very easily in a discovery studio. Go to structure, monitor, and intermolecular distance. You can able to predict this. This is a map output pharmacophore. This is after mapping the pharmacophore. Okay. Now, moving on, we are having the pharmacophore in our hand. And there are two ways. User created library. That means you are creating your library, and there are some default libraries are present in the database. Which one you want to use? If you have the different things like this, which method you want to follow? So if you have your user created library is present, then you can choose your ligand-based pharmacophore mapping or a stream library. Or if you're going to use a default library, you can use a search tree database. Or else, maybe you can use like this also. You have maybe lakhs of molecules on your hand, and you want to build a library. Yes, you can feasibly build a library in Discovery Studio. That will become a part of your software. From there also, you can go ahead with your screening of libraries. So this could be carried out in any way. That screening process can be carried out by means of a user specific or being a default base specific, or you can bring a database and you can build and you can predict also. So either way you can use this. So, but what is the actual requirement for this? So how do you create a library of compounds? So in case if you want to build a database, first you identify the chemical structures and search against popular search engines, chemical search engines like PubChem or Camel, or you can use a Zinc 15, whatever is it, or you do a research to get a compounds and input the structures in any formats. It could be in small molecular input line entry systems, we call it as SMILES, or you can use in different methods called an SDF formats or SD format, MOL format or MOL2 format. Any kind of molecules can be formats can be utilized, and you can do the structure similarity search. Once you do that, retry the compounds. If you have many molecules, it will try to download in a batch mode. That batch mode you can download in any format like SD, any kind of a chemical format like SD, SDF, MOL, MOL2, and use those compounds for mapping for the generated pharmacophore. So now one part of the study is completed. You generated the pharmacophore and you kept in your hand. But second step, you are trying to map these molecules with the pharmacophore to identify whether your molecules will have the potent activity, could have a potent activity like these molecules or not, that you can do only through the technique called mapping. So how do you do this mapping? Say for instance, you're having a pharmacophores being done. You go to the, if you want to do the ligand pharmacophore mapping, first you have to go to pharmacophores. Under the pharmacophores, compare pharmacophores and ligand pharmacophore mapping. So input the pharmacophore of your ligand and select the molecules. So what could be a ligands, or it could be a database like that, or which confirmation method you want to introduce, or which type of best mapping or in least mapping you can mention. Flexible method means you're giving a flexibility to your molecule. You want to omit any of these features, that you can mention any of the features also, or no, I just want to take it as like that also, you can do that. And if you want to screen your library of molecules, that is also feasible. You can select your molecules and then you can screen your molecules and you can check it out whether how the molecule should be mapped. In that way also you can bring up your input molecules. But here you can mention even the features. You can see the difference between each and every protocol. There we are not mentioning as much about your minimum features and maximum features. Here you can mention the features and you can tell the required features also to your systems. And then search query database. Here there will be already the input database will be there. From that input database, maybe your drug like sample, mini library, these databases are already a part of your system. And you can, as a part of your discovery studio, you can take even those molecules and you can go ahead with your analysis. So these are the three different ways there you can map your molecules and you can interpret your results. So ligand profiler, it is I told you, right, this is a most crucial feature of your software where you can map a set of ligands against a set of pharmacophores, and it uses a catalyst to map your input ligands to input pharmacophores. So usually you have only one pharmacophore. So far in the studies, we have only one pharmacophore and then the multiple ligands. So one pharmacophore, multiple ligands. But here in this case, ligand profiler, you can use multiple pharmacophores put together with the multiple ligands. Always your pharmacophore will be expressed. We have an extension for your compounds like MOL, MOL to SPSP. Even for the extension of your pharmacophore is .chm files. So it is called a catalyst query files. You can bring or even those files and you can build a pharmacophore. And using that pharmacophore, you can screen your ligands. So multiple compounds and multiple pharmacophore can be done by only using a ligand profiler tools present in Discovery Studio. So once you map your ligands, say for instance, I've used different pharmacophores have been introduced and been mapped with a different set of ligands. So what it will try to do, it will try to refit with the different molecules and see every time you come across these two things, confirmation fit, and then a features. So only these things will play a role in a pharmacophores. So which confirmation is good for fitting with this your molecule? Whether it is a good fit, you can see here for the best fit of the molecule, you can see the point vector feature for your acceptor, and then hydrophobic, ring aromatic, and then hydrophobic, and even in a positive ionizable. And for least fit molecules, you can see slightly there's a variations will be there, movements will be there, and some of the features might have been missed or not be mapped perfectly. You can see it perfectly it is mapped with this particular circle, but you can see here it is not mapped with this particular circle. That's why it's called as the least fit molecule. Good fit will have always a highest fit value. Low fit will have least fit values. And how do we give this ligand profiler? So it's just in the things I'm showing you. So you have to input your ligands. So how you could be a database of molecules also you can introduce. And if you want to introduce a database in advanced option, you have to opt for different databases and how many pharmacophores. Say for instance, this is a pharmacophore 1 I'm selecting for the cancer. This one for Parkinson disease, this one for malaria, this one for parasite disease, and this for bacteria, fungus. So like this, I have a different pharmacophores and set of molecules or drugs I download from the drug bank. So if I try to do this kinds of an analysis, we call it a repurposing of the drugs. That means with the different structure-based pharmacophore that you have generated, you are screening with, instead of doing a docking, just imagine that you have a lakhs of molecules. When you try to go for docking, obviously there will be a lot of computational cost will be taking and it will take run for a longer time. And then you have to interpret. And rather than that, if you have some sophisticated tools to do this process, rather than going for docking is also a technique where you have an identifying the potential shape complementarity amino acids and binding to the site and having interaction. Similarly, rather than docking, it is a mapping. That is the difference. The mapping is also going to do the same work. Wherever the features are going to present, it will go and match and gives you the results. And so using that, I can do the multiple mapping also. I can bring the, I pretending that I finish a structure-based pharmacophore priorly and having my ligands in my pharmacophores all in my hand, taking those pharmacophores and input human ligands. I'm trying to take different confirmation of ligands in my hand and looking for the best confirmation and then saving the energy threshold and keeping the scale fit values tools and align ligands to all these parameters I'm trying to set. If you're not sure about the input parameters, keep this as default and run the results. In case if you're not getting a results and haven't seen some difference in results, try to do this operations in your Discovery Studio or in these kinds of tools, then you can come out to draw the conclusions. So just given the molecules and just given the ligands and the different molecules with the different drug target proteins and different ligands, and you try to maps and get your results. So your results will look like this in ligand profile. One has been ranked by means of a pharmacophore, other by means of your molecule. How many molecules are fitting well with your pharmacophore? How many compounds are favorable to fit with your pharmacophore? Like this, you get a results. So interpretation is one means of fit value. Pharmacophores I have given. So this pharmacophore one is favorable for this particular drug. Similarly, this may be a target for cancer. This may be a target for an bacteria. Okay, fine. This molecule is showing a more fit value for the cancer with 1.02. Okay, then it could be taken for the further analysis. But the same compound with other pharmacophore showing 0.3. Okay, fine. Seems to be a moderate, but not good. So like this, if you are getting a more 1, 2, 3 more the number of fit value for this particular compound will have a can be utilized for the certain drug targets. That is how we have to interpret. So see the pharmacophore and look for the molecule and its fit value. It should be higher when you compare with the pharmacophore with the compound. Similar case, if you want to compare, they were molecule with the pharmacophore. So if you're trying to take one molecules, the same molecule is trying to map with the different pharmacophores or structure-based pharmacophore of the receptors. One cancer, okay, 3 is maybe a bacterial and 2 is maybe a Parkinson. Then comparatively, it is going well with the cancer. And similarly, the other molecule I'm trying to take it, when I see this molecule, okay, this molecule is going well to one other drug target, maybe a Parkinson is working out good as compared to a cancer disease. So just by looking into this particular results, I can able to interpret very interactively. Those are the molecules are good to fit with this particular drug targets and can be utilized for lead compounds or hits for further analysis. So like this, we need to draw the conclusion for your ligand profiler. This is a multiple interaction between your molecules as well as with your ligands. Once you're done, if you're not sure about it, if you're new to this technique, if you just want to learn about this technique, then you don't need to think about that result also. You will get automatically, once you click your results, you get a brief visualization of a heat map. This heat map is a tool will help you to identify by color identification. It's always better to interpret our results based on the graphical representation rather than a numerical or a word-based interpretation. When you look at a graph, you can see here, okay, fine. When even I am a layman, I'm not sure about it. So I can see here, okay, it's 0.3. It's not a good score. Oh, 4. Okay, 4. Wherever the 4 is there, okay, this molecule is fitting with a 4. Okay, these all are the compounds will fit for this particular drug target. These all the molecules will fit for this particular drug target. So these are the good. But as these molecules are not fitting for this drug target, so I can very well draw the conclusion like this by looking into this mapping. So these are nothing but a fit values. 4, okay, this will have very good interactions can be taken for a further optimization technique. So like this, even by looking at this kind of a graph also, you can interpret your results. So always you can see here, blue cells indicating a lower fit values, orange cells or red cells represent the more fit values. You can click and you can view results and you can draw your conclusion. Blind, if you're not sure about it, you can, by looking at colors, you can draw your conclusion. So these are the different methods and ways where we can introduce your even a ligand-based pharmacophore and structure-based pharmacophore, along with the interaction with a number of compounds or leads to come out with a fruitful results for any kind of drug targets. In a nutshell, I'll put together. So whatever we saw about it, in a nutshell, I could say that if we have a pharmacophore, basically what is required, we need a pharmacophore and need a set of ligands to perform this operation. And your superheroes are here, nothing but the chemical compounds. Only for chemical compounds, we are trying to analyze what are the features, and based on the features, we are giving a screening. From the screening, we are taking for a further optimization technique. So this is an overview of about ligand-based pharmacophore or a pharmacophore modeling into ligand-based drug designing. It's a kind of lead optimization technique. Thank you everyone for staying with me for watching this webinar. And if you have
Thank you,
queries,